What we're now understanding is that we don't entirely know about microbes in the womb, but we do know that the metabolites are crossing from an amniotic perspective, are coming from or into the fluid, and we don't know. And what a lot of people are starting to look at is how that is actually starting to shape the infant's microbiome even prior to birth.
Welcome to Commune. We are on a mission to inspire, heal, and bring the world closer together. We're in the river together. Good to be with you. Thanks for coming to our little commune up in the mountains.
Thank you for having me.
I always say,
again.
Yeah, I always say, when you feel like you're lost, that's when you're almost here. So I don't know if you felt like you were lost on the way up here, but
Wasted.
oh, good.
And by transference, I did.
So you hold the dubious honor of being my wife's favorite interview. Wow. And my wife does not suffer fools.
I noticed that day I met her on Zoom. I had a feeling she does not. That's an amazing honor. Well, you have the honor, and I say this all the time to people, when we met during COVID, during COVID still is here, but at the height of kind of lockdown, it truly, and I'm not saying that just because I'm here, really my most memorable and favorite podcast I ever did. So, which is why I wanted to take the extra time and prepare properly for this.
I appreciate that, because you are my go-to for stringent science. Mm-hmm. And I'll probably pepper you with some true-false, to the degree that there are true falses out there, because there's mostly probably nuance. But there are a lot of claims that get thrown around loosely in the world of the microbiome and probiotics, et cetera. And some of those claims are very well intentioned and super exciting. And yeah. I've probably been guilty of a claim or two, mostly just because I'm playing catch up. I'm not a doctor or a gastroenterologist. I'm just reading as much as I possibly can with great curiosity. And this field that you've gone into is, I think one of the reasons why it's so exciting is that it provides so much agency for people,
yes,
including myself, because I suffered from, I guess, what you might call dysbiosis, and it had all sorts of really negative knock-on impacts in my life. And it took me a long time to literally get my act together. And part of getting that act together was learning about what you do, and then being able to apply science and, more specifically, mechanism to my own life. So I'm grateful for that. And I loved that interview too. And in prepping for this one, I went back to that one, and I was like, oh yeah, that was good. So I'd love to start, with some history of our human relationship with bacteria, because we didn't know about microbes forever. And particularly as it pertains to our understanding of disease, I think back to the Black Death, for example, and the prevailing theory at that juncture was this notion of miasma theory, that there was this kind of pestilence in the air, and I think there was
noxious air.
Noxious air.
Yes.
I think there was an element of the stars or the planets aligning too, but. Yes. But we progressed significantly since that point, and discovered microbes. And at this point, I think it would be more appropriate to pass the mic, because I think it would really be helpful to set the table to get a little bit of our background of our tawdry and ambiguous relationship with microbes.
Well, it's interesting that you said, it's interesting. I've been listening to you when you said we didn't know a lot about them. One of the things I think that's come out of the last decade, and like remembering our place as humans, but also our place as like the Western narcissism, that like our lens is the lens. One of the interesting things I learned recently is that even the Lakota have a word for microbes because there are a lot of cultures around the world that always understood. For example, the Lakota use this word for what translates to little monsters, and how they had known for so long to never drink from still water, as an example.
Really?
Of course, that's where
Sure.
microbes can proliferate. There's not a lot of movement. Temperatures can be optimal. The acid conditions can be optimal. So one of the things I always try to remind myself when I talk about these things is that, it's like history is written by the victors.
Yeah,
And in
sure.
science, history was written by a lot of white guys. And then so I think that there's a lot of evidence to suggest that, other cultures certainly had started to understand this otherness in different ways. Even prior to the white guys.
Yeah, maybe even TCM to some degree.
Absolutely. And so, and in different language, different ways of thinking about it, but I do have to believe that, the notion that, Ayurvedic frameworks were looking already at these types of digestion as a means to understand the way that your body processes like illness, pathology, good, food, etc., other information. And then TCM, of course, like looking at your tongue, for example, where we know now that it's, for my world, it's called the oral microbiome, and one of the niches of the oral microbiome. But of course, in TCM, the first thing one of some of those practitioners will do is look at your tongue. So I think that just before I get into like the white guy history of microbes, which has a very black and white and othering way of looking at science, health, and our bodies, it's just important to recognize that my guess is that though not well documented, there were a lot of cultures and frameworks and ways of thinking about the body, health, the environment, where they probably already knew of some of these other organisms that were not human or simple organisms. So, but in terms of just the way that this kind of like antibacterial or antimicrobial world came about was definitely from, and I think until recently we never really experienced this, but throughout history we now die of non-communicable diseases more than anything else. So we're used to a world where heart disease, type 2 diabetes, these conditions that you can't catch, unless of course you think poor food systems and these things are contagious, which that's a whole other podcast.
Yeah, we could make an argument there.
But for the most part, you're absolutely right. It's certainly the Black Plague and the notion of miasma and like noxious air, that something's in the air, which by the way probably, and we know this from molecules and the way that particles and the way that even just COVID hung in the air and the way that some of those things worked absolutely was probably partially true. But when germ theory came about and Louis Pasteur, and they started to really understand the connectivity between very specific microbes or germs and their relationship to pathogens, you have to remember it was at a time where they weren't thinking about like the soil losing nutrient density, so they were only thinking, wow, thousands of people, millions of people are dying from these things. So how could they think that there must be good ones, because that was obviously the construct that was happening and what they were able to correlate it to. And so Louis Pasteur, which of course led to pasteurization and some really important discoveries, I think started this idea that these things that you can't see, that he was able to correlate with the specific diseases, was the cause of them. And of course, in history, when you think something's the cause of something, you immediately, there's an assumption that whether there was no question and not a lot of questions asked as to whether or not there were good ones, it was just, well, now how do we eradicate them? And you have to remember, like, the world you're in, right? Which is, othering was part of the way that you derived power.
Well, certainly the anthropocentric view of the world, we went out and killed all of the things that were bigger than us.
That's right.
And we ran out of those predators.
Or that looked different, or that we needed to exert power over.
And then what was left? Well, these things that we couldn't see, these insidious little...
But that were killing people, in some really bad, I mean, obviously that we know about the big ones, but there are all kinds of, and it was at a time, sanitation wasn't what it is, hygiene isn't what it is now, water systems, et cetera, were of course very different. And so, and I think that really led to, starting in like the like mid to late, 19th century, and then going into the 1900s, like an entire wave of, Joseph Lister, which of course led to like Listerine, and this notion that, and you have to remember there's some religious and puritanical ideals in here too, which is cleansing one. It's getting rid of the bad, right? And so these led to this kind of obsession with hygiene and this notion that these things were dirty, and that they cause disease. And of course, that led to many really important things like our sanitation systems, which did reduce disease, washing hands after surgery or before surgery, which led to the saving so many lives, led to really important things like antibiotics, which in the right moments, not the 251 million prescriptions written in the United States every year necessarily, but in many instances, like they are one of the most important inventions we've ever had.
Yeah, I mean, I read some statistics about World War II that were just astounding. I actually read something that might be apocryphal, but that the United States had antibiotics to administer during World War II, and Nazi Germany didn't.
Interesting.
And this was some sort of delineation there between how we could save our soldiers. But I don't know if that's true. But it certainly fits the right time frame.
Well, we've certainly made up for it now.
Yeah. Well, it has become, proliferated as part of a whole paradigm, a whole approach to beyond human physiology, right? So
absolutely.
It's how we treat soil, et cetera.
Absolutely. I think it led, and I think what it did, look, fear is, and I think we only have to look to the last couple of years to see how it impacts our world at scale and is currently when you look around the world. But fear is very powerful, and it creates irrational spectrums that tends to be very polarizing. And we certainly as a culture went in the direction of kill them all, and I think we've done that in a lot of areas. And of course we've done the same thing with, and we're also very short-sighted as a species, right? We're very one of our favorite scientists, George Church, who's runs the Wyss Institute at Harvard, who's known for, among many other crazy things, wanting to bring back the woolly mammoth, introduce the DNA into the East Asiatic elephant to be able to begin punching holes in the permafrost to be able to reverse climate change. So he's an amazing thinker. But he just says, as a species, we just can't plan ahead. Yeah. Which is absolutely not the way that we work. And so, of course, that eradicating all the bad led to pesticides. I mean, it's the same thing.
We don't think of planning ahead as practical, which is strange. We think of like business people as very practical.
That's right.
But they're looking like 3 to 5 years.
Yeah. 3 to 5 years, exactly.
I mean, I think of like the polio virus, for example, and the national fervor to eradicate the polio virus and the March of Dimes. And, I think there was either 1.2 or 1.8 million children, many of whom were in the control group, but basically whose parents just signed them up for the polio vaccine trials. That's right. And you're like, what? I know. And so, this commitment to excoriate and eviscerate microbes had some positive impact. So we did a pretty decent job the last couple of years notwithstanding of eliminating infectious disease, but we traded that in for something.
Yes, and I think that's just because, to your point earlier, like there was no acknowledgement of nuance, and there wasn't really enough curiosity to think about, well, what if there's good ones that we actually like really need? To be fair, we didn't have a lot of the technologies to know that. In some cases, so it would have taken, although there are scientists, and this is why I say it's important to know who the history is coming from. Carl Sagan's first wife, who's one of my personal heroes, Lynn Margulies, who people don't hear enough about. I highly suggest you read everything she's ever written and done. She posthumously, and should have won probably the Nobel Prize during her lifetime, posthumously was recognized for the endosymbiosis theory, which is actually the theory of how complex life started. Which is the notion that, archaea ate a bacteria, or bacteria ate archaea, which that's still a chicken
Yeah,
chicken
right,
egg
yeah.
conversation in science, but she was able to identify that like mitochondria, the structure of simple cells, resembled enough
Wait, that's
the
her
structure.
theory.
Yeah.
Get out of here.
Yeah.
That's
So,
incredible.
and she was never recognized for it, and she died of cancer fairly early. Of course, he gets lots of credit for things, which he should. But she was definitely at a time where women in science and STEM were just not recognized. And so there was a lot of things I think may have been known that are being known now and kind of claimed. But to give full credit from a scientific perspective, I mean, a lot of these things could not have been known in the way they are today. And one of the things I love, there's a book I was going to bring you a book. There's a book I'm gonna bring to you that I gift to everybody. I think I've bought every vintage copy that exists in the world. But Horace Freeland Johnson, Judson, who is a science communicator and translator, put together this like kind of beautiful book. And one of the things it says about science is that, you know, people don't acknowledge, and of course this happened during COVID because it's a really hard, nuanced thing to understand, is that, you know, there's scaffolding that happens in a field like this, right? Mm-hmm. It's like the people who are creating like calculations for physics, they weren't trying to go to the moon. They were just calculating and making calculations that you needed then years, you know, decades later to go to the moon. And so, you know, it wasn't until the Human Genome Project that we had like 16S sequencing, for example, shotgun sequencing, where then then you could start characterizing the DNA. Then you could start characterizing the DNA, not just of human cells, but of bacterial cells. You know, and so, you know, these things had to kind of progress along with also what we could know. And so, you know, in some cases, it's easy to look back and say, like, how could they? Everyone just wanted to kill everything. And that certainly, I think there's a lot of religious, societal reasons why it contributed, fear, what happens at scale in trying to communicate to people at any level. You know, as we've seen in the last couple of years, like in mass is really challenging, let alone the entire world. But you also have to recognize that like sometimes science can't know something if you don't even have a tool, you know, sometimes to know it. And so I think that that's part of what has really accelerated the field over the last, you know, over a decade now, 10 to 15 years.
Yeah, I think the Human Genome Project is an interesting inflection point maybe, because so we had Darwin, and then we kind of had the Watson and Crick paradigm of the world in the early 50s, and we put a lot of stock in human health being centered around the human cell.
Yeah.
And then all of a sudden we could sequence the human genome. And maybe can you pull on that thread a little bit, like what happened?
Oh, sure. I mean, it wasn't, you know, the Human Genome Project wasn't looking at microbes necessarily, but what it did was allow us to get the technology and the kind of sequencing technology so that you could start to understand, like, well, when you're starting to look now at all the other constituents of the human body, you know, and then you had, like, under the Obama administration, the Human Microbiome Project, which used that technology and really, and it was, I think, one of the largest initiatives ever done, which is $173 million, which for the United States doing, you know, at NIH level is like a big grant. And that was really to characterize the human microbiome and try and answer the question, like, what is a healthy microbiome?
Yeah, well, I bring up you brought up the Human Genome Project, but I comment on it because the result was somewhat surprising. I think we had a preconception of humans being a little bit more sophisticated than we are.
Yes, correct. You mean in terms of our genomic redundancy with like bananas?
Yeah, exactly. I keep seeing the number being modified, but somewhere around 20,000 genes or something.
Yes.
And so then the question was, well, how are we so complex, or how do we how does our DNA code for as many proteins as it needs to with only 20,000 genes? And I think, and this stumped a lot of scientists for a while, but potentially, and this is actually my guess, but my guess is that opened some of the doorway to the study of microbes again.
And certainly on also how it opened the doorway for how that could be engineered or modified, right? Which is kind of what's happening now. Where I think a lot of the, like with CRISPR and a lot of the new technology, and then of course the CRISPR's kind of even old, not old news, but there's so many more CRISPRs now, and you know what we're gonna do with that. But you know the hard thing, you know, there's a stat that gets thrown around in microbiome a lot because, you know, microbes, I think at one of the last counts express, you know, over 300 times the amount of genes than our human cells. But that gets thrown around, and people are like, see, it's even more important. And you're like, yeah. My co-founder says I'd still trade some of those human genes. I'd trade a bunch of those microbial genes in for some of the human. They do some pretty important coding. So, so it's not a one-to-one or apples to apples, but it is interesting to think about that from a cell count perspective, at least as of the last Weizmann Institute paper that kind of approximated in the human body that human and microbial cells were about 50-50, which is different than the 10-to-1 that got, I think, thrown around a lot prior to that. But that's unto itself the idea that you're by 50% of your body cellularly or is not human.
Right, and that would exclude even the archaea bacteria mitochondria dilemma.
Yeah, right, exactly.
But and so I've seen 38 trillion. That's the number I see popping around.
That's probably the number. That's the count that it's a citation that we use for number of bacteria.
Okay.
But the one that we use the most right now is about that 50-50. Bacteria obviously constituting like large majority of the microbial population, but then of course there's fungi and viruses, and there's the virome, and there's the metabolome, which is a whole other thing, which is not the microbes but what they're making, which is probably where the field is headed.
So they have their own classification now.
They do.
Okay.
Yes.
All right, we'll get there.
Yes.
So about 70 trillion, give or take, cells in the human body, about some 50-50 relationship or proportion between human cells and microbial cells. And where are most of them?
Well, the Human Microbiome Project estimated that, the majority of them, and probably why the field started where it did and why a lot of people kind of conflate the word microbiome with like the gut microbiome or gut health or any number of the marketing alliterations of microbiome are primarily in the gut, in the GI tract, and that is where there's the largest density of them. The Human Microbiome Project put that somewhere between 500 and 1,000 species. We've seen bigger numbers than that. After that, the oral microbiome is the most diverse, not necessarily the most microbes, but the most diverse. So about 700 species in the oral microbiome, each with like very specific niches. So the tongue has a different microbiome than like the cheeks. The saliva has its own microbiome. So, but as a whole, the oral microbiome is about 700. And then you have, in women, the vaginal microbiome, there's the genital microbiome. There's the skin microbiome, which again is not a lot and actually is less and less diverse than the gut and the mouth. And then, of course, there's these smaller niches like the optical microbiome, the nasal microbiome, which I think we kind of got some wind of during COVID, to understand that, of course, microbes interact with your nasal passage. But of course, there was a lot of talk about whether or not the nasal microbiome predisposed you to long COVID, to susceptibility, even contracting COVID, because they would see roommates that would not, the other one wouldn't get COVID, and looking at the nasal microbiome as a potential, or any of the airway microbiomes as a potential predisposition to contracting or protecting against. So there's kind of, and then of course there's the scalp microbiome. And so really like anywhere where your body touches an external surface or there's information coming in, there's usually microbes. And then some sort of niche, the axillary microbiome, which is your underarm, your armpit, for example.
Right.
And then even on your skin, the microbiome of your face will have a different composition than the microbiome of the chest or the back, for example, which is why you'll see like specific conditions like specific acnes or whatever like only happen in certain areas, of course, because of some of the environmental conditions of those. But you can imagine from an evolutionary perspective, some microbes evolved to be on that part of the body. And so dysbiosis in that part of the body would be very different than like the palms of your hands, where actually it's not a very substantial microbiome.
Right. I think I read an article in the New York Times about the armpit microbiome. And I think the claim was that sweat or perspiration doesn't actually have an odor in and of itself, but when you sweat in your armpit, it is actually being essentially metabolized by the microbiome, and that's actually producing a particular kind of odor, such that our attraction to another mate, when I've done it from time to time in the laundry room, picked up my wife's like yoga shirt and been like, oh, I still love her.
Amazing. That's so funny.
That our microbiome might be fixing the game a little bit in terms of who might be the best mate based on the attraction of that odor. So
well, what they produce are called VOCs, volatile organic compounds, and those are produced in other parts of the body beyond the armpit. That is what it contributes to halitosis. So it creates the same thing, microbial, metabolites that are coming from microbial activity that create halitosis. Then there's really interesting ones that even just come off the feet. Such that, yours is a sexier example, but just to show you that they have trained dogs to be able to smell the volatile organic compounds and be able to identify children with malaria, for example.
Wow.
So thinking about VOCs, yes, as an attraction, and certainly more fun to talk about when it comes to love.
Well, I might have a foot fetish.
Yes,
you don't know about
totally. But also, if you think about it as a diagnostic too. People say cancer I remember my mom had pancreatic cancer. You could smell it. And so it's really interesting to think about when there's dysbiosis, so when there's something disturbed in a specific niche or microbiome, how that could be producing volatile organic compounds that create smells that actually could lead to interesting diagnostics too, which is kind of interesting to think about. And that was what I was saying, a lot of the field is also moving towards not just which microbes are there, but what is their function, what are they doing, what are they making, what are they creating, and what is that telling us?
Right. Okay, this is a perfect bridge into defining a few terms that I think get really confusing for people.
Sure.
So there's prebiotics, there's probiotics, and there's postbiotics. And can you help disentangle that morass?
Sure, and I'll give you kind of the nerdy ISAP version, which is more the International Society of Prebiotics and Probiotics, who regulate the term scientifically for the international community. I will just preface this by saying that scientists misuse the term all the time, and in fact, we wrote a paper. Dr. Gregor Reed, who chaired the panel for the UN in 2001, and then has done all the subsequent definitional updates for the WHO of the term probiotic. And he was president of ISAP, and he wrote a paper with my co-founder, a number of other scientists, for Frontiers in Microbiology, I think 2-3 years ago, having to just re-clarify, not for the wellness community, not for the people shopping in grocery stores who are buying probiotic tortilla chips, but actually for scientists, because there's so much misunderstanding. And so hopefully that's also comforting for people who don't understand it, because there's a lot of misuse of the term, even honestly within the scientific field. But the term, to start with, I'll start with a probiotic, because it's you can work from there really easily. So probiotic, according to the definition, is a live microorganism when administered in an adequate dose confers a benefit to the host. And if you just break that down...
You've never said that before, right?
I've written that. Yes, I think all of us at Seed have it tattooed in our... It's ingrained in our memory.
Yeah, it's on a dusty scroll.
And I can't actually recite the definitions for the other two, but I can tell you what they are. And what's important, if you just break that down, is it's live, which means that you have to demonstrate that it's live and viable as an organism. It's administered in a dose, which means that through some aspect of human trial or clinical research, you've derived what the appropriate dose is to be able to effect a measurable benefit in a host. And that could be a dog, and it could be a human, it could be a honeybee. But that creates, as you can imagine, some precision to which microbe, which means you can't just say, the species. You need to know what strain it is. You need to know which microbe it is. You need to establish the aliveness of it, and then the other pieces. And that is established through research, rigor, and then obviously the ability to reproduce that dosage and that effect across, depending on what you're measuring, either a heterogeneous population, a specific disease population, for a specific indication, et cetera. And so that's why the term itself, for example, in the EU or places like Japan, where there's really stringent regulation, where they adhere to that term from a consumer perspective too, the term probiotic cannot be used in Europe, for example, without the adherence to that definition. In the US, the term's not regulated, which I think is probably why you and I could go on Amazon right now and buy probiotic pillowcases and cleaning products, tortilla chips and chocolate bars. But that is, and any number of other foods and beverages that might be fermented but may not meet the definition.
Right. So it has to be live bacteria, and then I assume in order for it to confer a benefit, it actually has to be able to make it through your stomach and all the acids and enzymes that are going on in there, and then through your small intestine and into your colon.
I mean, look, there's nuance to this, right? Yeah. One of the reasons we believe, at least for Seed, that our kind of flagship product, DSO1, that does have a really interesting delivery system, it's a symbiotic, so it's a little bit different. Why that's so effective is because we're able to understand the release profile through the GI tract, and we do think that's part of why it is so effective, in addition to the dose, and in addition to the strain, the consortia of strains that were chosen for very specific endpoints. That is not to say, and that probably leads us to the definition of postbiotic, that is not to say, and there is research to suggest, that postbiotics, which do not have to be live, they can just be dead organisms really, or not necessarily be viable as live organisms, can also be administered. And there's signaling that some people believe happens, and I think there's been some early research to this effect, that just taking dead microbes may also have a benefit, because there's signaling that can happen as they move through the GI tract, even if they're not alive. It's not the world that we spend the most time in, but it would be unscientific to dismiss it. Yeah. And I think there's some work in postbiotics that's being looked at in other biomes of the body, like skin and other applications, where there may be viability. It's just doesn't make it a probiotic, right? Just to be definitional about it, and it doesn't necessarily. It from what I understand, it's been hard to understand what the mechanism of action is, and how it's working. But it doesn't mean that they couldn't be effective.
Yeah, we have a great sourdough culture, for example.
There you go.
And then we bake it.
Yes.
And you're like, well,
exactly.
What happened?
And it still could be delicious. And there's also, you have to do it in a randomized placebo-controlled trial. So you need to know, is it statistically more significant than placebo? So I think it would just depend. My answer on that would just be for what, for how is it being studied? What is it being used for, claimed for? I think what a lot of the industry, particularly I've seen this in skincare, where there's a lot of class action suits right now against people that were using the term probiotic on their skincare. They're moving to postbiotic, because then you can just use dead lysates and throw them in a moisturizer and call it a biotic moisturizer, and people think that you're saying probiotic because they don't know. Or they're saying postbiotic as if it's they don't know what it is, but it's a new technology and it sounds effective.
Sounds effective. But when we're talking about really beneficial postbiotics, are we mostly talking about short chain fatty acids in your world?
The postbiotic technically is really the dead microbe. I think a lot of people conflate it also with, and I've seen it used interchangeably with, and again, even in science, with the metabolites that are being made by microbes. Okay. But as far as the ISAP definition, doesn't actually.
So there is a delineation between postbiotics and metabolites.
As of those definitions, yes, but I have seen it used, as I said, even in science, a bit interchangeably. And I think a lot of that will be much more clearly defined, and particularly as the metabolome and the metabolites themselves become so much more important as we look at future of human health, as we look at new drugs, new therapeutics, new ways of understanding, like new biomarkers to understand like whether or not, as I said, so much of the field is moving towards not so much like what's in your microbiome, but like what they're doing, because microbes have a lot of what they call functional redundancy, which is that two different microbes can make the same thing. So looking at which ones are there is not necessarily as important as how much butyrate is being produced necessarily, or like as a short chain fatty acid,
Right.
as one example. And then prebiotic, which is I think the last one, is a substrate or compound that's kind of selectively utilized by microbes to do a few things. One, it's defined as like it's a compound or substrate that's selectively utilized by a very small, like specific niche of microbes to either proliferate, to grow, so to use it as like a food source. So prebiotic is not an organism at all, just to be like super clear, because that can be confusing. Prebiotic is really like, it's the most, if I explain it to a fifth grader, it's like, it's food for your microbes, or a really important compound that microbes use to make other things that are really important for your body, is the way that I kind of explain it to like kids.
Yeah, so we hear about like polyphenols, fiber, resistant starches.
Yep,
Would those all fall into the prebiotic category?
There's some nuance to the way that they can be categorized, but in general, I think that's a really kind of good way to really think about them. There's a lot of, I think another point of nuance with prebiotics is whether they're fermenting or non-fermenting, which is kind of interesting. We work with a compound in our adult product that is non-fermenting. And so for a lot of people that may be listening who, in your community, who are on low FODMAP diets, or where prebiotics can be, that fermentative activity can be super uncomfortable. Like people who can't, you take inulin or certain like FOS or GOS or like these long chain kind of saccharides, and some of the shorter chain ones that cause fermentation at different parts of the GI tract. Though so there's a nice distinction between like fermenting and non-fermenting. And so we work with, for our adult product, non-fermenting, which is a compound called punicalgin that has been tied to the production of a really interesting metabolite called urolithin A, which like a couple of years ago got like, I think LA Times was like, this is the fountain of youth. So I think we're going to start to see that what they're making, and just to give like a preview of some very exciting news we'll probably be announcing in the next few months, is that what they're making will start to move us away from looking at like stool and maybe more into like the blood, because you can look at the metabolites circulate into the blood and other parts of the body, and it gets us a little bit away from the what we call kind of the Friendster of microbiome, which is like just sequencing the composition of microbes in your stool, which if you have a parasite or you're looking for something like that is really important just to understand. But it's not the end all be all of where microbiome's going.
Got it. And how long does the average microbe stick around in your gut?
It's a great question. Well, in terms of the ones that are residents that live there for a long time, in some cases, obviously after a course of antibiotics, my answer would be different. After a binge drinking for 7 days, my answer would be different. So a lot of that has to do with, one of the interesting things about the microbiome is that it's incredible. You were saying that it creates agency, and part of that is because it's so malleable, right? Like part of the lack of like that exhilarating kind of climax of the Human Genome Project movie is that you can sequence the human genome, but like you don't go to Whole Foods and buy anything for human genome. Like you're like, and I can't do anything about that. So and not to say that there are certain areas, of course, where like epigenome and certainly environmental factors impact a lot of things. Just because you're predisposed to something genetically doesn't mean you're gonna. There's so many factors that you can have an impact, but you're not actually changing your genome. You're just either you're changing whether or not something may express itself that you're predisposed to, and having an impact there, which is different than the actual like microbiome, which I think that the last stat or the citation that we had looked at again from the Weizmann Institute, I think from Eli Elinov, who's another like extraordinary microbiome scientist in Israel, who says, I think he had cited only about 2% of our microbiome is probably genomically informed, and 98% comes from information, literally, like the environment, like exposure, choices you make, choices you don't make, but you're exposed to. And so you can just imagine how much like agency that provides over the course of a lifetime.
Yeah, and so, I mean, how much agency, I guess I would ask, does one have to swap it out, for example? I mean, I've seen that the life expectancy of a microbe in your gut is 10 minutes or 20
Yes,
minutes.
yep.
You know, so if I'm thinking, okay, well, there's 38 trillion of them in there, have they all changed since we started talking? I mean,
Right. Oh, I see what you're saying. Yeah, no, I mean, after about 3 to 5 years, I mean, it's a really great question. I can answer it from a probiotic perspective, which is that the administration of newer microbes, if you stopped taking a probiotic, you would see them kind of wash out within 3 to 4 weeks. And actually, a lot of people who are changing a probiotic or making a big dietary change, a lot of people will say give like this kind of washout period of somewhere between 2, 3, 4 weeks. And then after that, you can kind of no longer see those microbes present in, at least looking at stool, for example. And that's important to know just because a lot of people think about probiotics as this thing that you take once and then it like restores, rebalances, which by the way, there's no one who wishes that's how it worked more than I do because it'd be so much more easier to explain. But really the microbes that you're taking are transient. They do their work on the road. They come through, they're signaling, they're interacting, and then they're leaving. Yeah. And so they're not as, they're not like full-time residents, which would mean that they're colonizing. Which is kind of yet to be seen. And I think there's nuance around, as well as no one in at least the serious probiotic world believes that like a microbe has to colonize in order to quote unquote work, meaning to do what it was designed or studied to do.
Yeah. So it's like a whirlpool that you can go, you can recognize its form,
right?
But the actual molecules of water have moved on, or a flame.
Yeah, well, you can recognize, you'd also recognize what they made while they were there or helped make, which is kind of one of the, why I was saying that function is going to be so much more important than composition.
Mm-hmm. So maybe take a moment and explain gut health. And thriving efflorescent gut health versus dysbiosis and maybe even downstream from there, intestinal permeability.
Sure. I mean gut health can mean the challenge. I think I can tell you what I think people mean by it. Yeah. And I think that for the most part, I think there's this general consensus, although like a lot of things, I think there's always a lot of evangelism over evidence, and a lot of people are always like further along or ahead than where maybe the science is. That being said, I think there's a lot of consensus that the gut and the related axes, whether it's the gut-lung axis, the gut-brain axis, the gut-liver axis, the gut, of course, the various understanding that the gut has this connectivity and impact on our systemic health is, I think, fairly well believed and characterized and understood. Of course, we're starting to understand all kinds of new axes and mechanisms and pathways by which microbes make an impact, and or the gut as a whole. And of course, from an immune perspective, I think we're just starting to even understand like incredible correlations with like childhood allergies and very specific just with a lot of nuance and understanding at like the mechanistic level of exactly how this all works. However, there are still really complex conditions like autoimmune conditions and things that are so multifactorial that are like, just challenging and complex. But I think when people say gut health, I think there is this notion of this feeling that this ecosystem is functioning well, such that systemically you're functioning well. And I think that of course starts very locally from this understanding of digestion, which I think, for a long time, I think as we were growing up, and particularly in like the antibiotics generation where like parts of our health was so siloed, right. I think digestive health was something that was just seen as like, are you constipated Or do you have gas? Do you have, do you need to just like start pumping antacids basically? But I think now we're starting to understand that digestion is a real biomarker for like a lot of other things that are happening.
Yeah.
And I think that comes and stems from our belief that if this is healthy and functioning, there's a lot of other things that are healthy and functioning. And I think that is a beautiful new framework. Again, whether it's sometimes a little bit sensationalized in terms of some of the specificity, I still think it's a better framework than continuing to look at so much of the way we looked at things in such a siloed way, in like our parents' generation or as we were growing up. And then in terms of dysbiosis, I think, there's a few different definitions of dysbiosis. I think in general, it's like the easiest way of thinking about it for people who don't know the term is like just when things are out of balance in the microbiome, which I say that with massive quotation marks because we're starting to understand that we don't entirely know what that means yet. I think we thought we did because we were so focused on like what was in there. But now that things are moving to like, well, what are they doing? Just out of balance, that understanding scientifically may change. But in general, it's easy to think about overgrowth of the bad stuff. So too much room in the neighborhood for the bad stuff to move in, basically, and kind of take over. I think another way to think about it would be like just total like loss of microbial density, not even diversity. So after a course of antibiotics where it creates a very opportunistic field where it's unclear who's going to win, who's going to take up the most residents, and is there going to be like a good balance after some of those more like indiscriminating like wipe out everything moments, which by the way in some cases are really important, particularly in the case of a really bad pathogen or something.
Yeah.
And so those are really, and then there's this notion of just kind of being out of balance, which means that there's just very disproportionate amounts of different microbial communities. And what that leads to, depending on how that dysbiosis is happening, is an unhealthy amount of permeability. There's always some aspects of permeability, but when the junctions or the epithelial wall and there's this allowance of things to flow too freely into the bloodstream and out of the GI tract, and that's out of balance, that's where you start to see inflammation and of course the body's response to those compounds that are not supposed to be going out of the GI tract and into the body, and that's where you start to see autoimmune and conditions, responses, etc. And obviously like just misfiring of the immune system because it's things that are not supposed to be outside the GI tract.
Got it. That's helpful. So I assume there's a lot of contributors.
Yes. Many.
To this condition of,
many.
Well, a lot of people call it leaky gut, but intestinal permeability, the separation of the tight junctures in the epithelial.
Yes.
And can you take us through some of the primary contributors?
Yeah, of course. I mean, I always say this is kind of the boring part because it's so much of like what people know for health, but at least now I think research in microbiome is at a point where like we really do know that like these compounds like hurt and or nourish or help in a healthy individual. So I think, we talked about antibiotics, but, again, I'll just stress that like in the right moment, they'll save your life. So I'm not anti-antibiotics. I would also say just for so many people who are more in like the more wellness and alternative or functional world, that just because oregano oil and colloidal silver and garlic seem quote unquote natural, that the overuse of those are really powerful antimicrobials, and just something to really like, oregano oil is used as an antibiotic in livestock. Like these are really strong antimicrobials that should not just be just indiscriminately taken at the first sign of a cold just because you don't want to take antibiotics because you are taking a very strong antimicrobial.
Penicillin is a fungi.
Yeah, of course.
Right?
Yeah,
So
well, and aspirin is willow bark. So, So that's I think one of my favorite books, which is, if you know the writer Alan Levinovitz, he wrote the book Natural. You would love his writing. He wrote The Gluten Lie. Okay. Yes, probably that and Natural probably don't make him a crowd favorite, at yoga retreats, but it's a really important perspective. He writes about like the origin of vanilla and like, how corrupt that is, and the orchid industry, and just like our perception of what's natural. And just, like you said at the beginning of this, just the importance of asking questions.
Yeah. Okay, so...
Yeah, so antibiotics, the reason I was bringing it up is, just to mention the fact that like some of those more natural antimicrobials are very powerful. Second is that unless it is a known infection of bacterial origin, or you're doing it under some sort of very specific protocol either prior to an FMT or prior to some sort of other intervention, I would just caution against like using antibiotics indiscriminately. I think a lot of people like start to feel a cold and they're like, oh, I have a trip coming up, and they just like get a Z-Pack just to be sure. And so I think not only is that leading to antimicrobial resistance, which is probably a whole other episode, and the downfall of humanity, but it is definitely something that can impact your gut, and then of course other functions in your health if you don't need it and you take them indiscriminately, which a lot of people do. The second is alcohol, where we're doing some really interesting research now, just looking at the impact of alcohol on the human cell wall, as well as the microbiome. And I think some of our early in vitro data was just on like 2 shots of Grey Goose vodka in this like really cool model called SHIME, which is like a simulation of the human intestinal system. It's a research partner in Belgium or in Brussels, I believe. And that's really interesting
so
because
they can kind of simulate the digestion system in a lab, basically.
Yeah, exactly. It's a chamber. It's like the size of like maybe half this room, and they have every chamber, and they can control the microbial population. They control digestive acid, bile salt. It's incredible. You can like simulate human digestion.
Yeah, I read a book recently that this is very crude, to be honest. It was, I think, in the 19th century, and there was a furrier or fur trader, I should say, who got shot in a duel or something like that. Something very 1800s. And there was a physician that saved him, but then he couldn't be a fur trader anymore, so he went to go work for this physician, but he couldn't suture up the actual wound. And so the physician ended up using him as as an experiment. Amazing. And he would tie what I guess would be the equivalent of like fishing line.
Yes, I do know what you're talking about.
Onto food and actually stick it through his digestive system.
I know what you're talking about. In fact, they simulated it in a movie that I saw when I was a child, and like never forgot that at all.
Gruesome.
It was like crazy. It's crazy. Yeah, it's incredible. It's in some movie somewhere, and I absolutely remember that. Yeah, it's an amazing story. And by the way, that one thing, just to go back to the beginning of our conversation that I did forget to mention, we were talking about at the beginning of all of this, is that some things, and like, I'm always so surprised by this because I'm just like, really, no one ever just like went in there? But some of the reasons we don't know about things is because we couldn't get to them. Yeah. So like, obviously like the bottom of the ocean is a great example, but, and how much we don't know about that, which is like an amazing amount of unknown knowledge given what we know about like space, for example. But the gut is really, unless you're doing like punch biopsies or incredibly invasive testing, it is very hard to get to it, to understand it.
It's deep ocean.
It's deep ocean. And so in a lot of ways, I forgot to mention that earlier, just because like sometimes you just think of the human body as something that they used to like just like open up and check out, but actually like in a lot of ways, like sometimes research is just stalled because there aren't always these like really either ethical or like kind of easy ways to get to a location in the body, and that it just ends up being like not studied as much as like your skin.
Yeah. So you're gonna kill my dreams on resveratrol and red wine now?
I'm not going to kill any dreams about no, actually, because I think, and that's where the nuance comes in, right? So it's like, okay, so alcohol is not great, but if I am going to drink it, red wine obviously is like higher polyphenolic count than, and obviously other compounds that, despite sometimes questionable scientific histories, can be beneficial. And so, so I think a lot of it then just comes down to, how much and which one and how often and with what. But I think for sure, like, I think in general, just like the moderation of alcohol intake, knowing that it does impact the microbiome and it does impact the human cell wall too, in terms of a permeability perspective is really interesting to like at least just know about. And also, again, the not doing, it's then, okay, well, what are the things you can do to nurture and kind of nourish that ecosystem that could at least help temper some of the the things you really still want to do.
Yeah. So I'm going to draw you into the animal protein debate for 1 second. So what's
Okay.
up? I've read a lot about TMAO and all
Yes.
these other kinds of things.
Yes.
Any particular viewpoint on it?
I mean, I think. Well, I'll say actually, it's really interesting. We're doing some research on whether there's a meat microbiome, and looking at the microbiome of meat actually in farm raised versus factory versus, and looking at some of those, and also a lot of the research recently has demonstrated that the differences are not as stark as I think we would like to
Really?
see, not from a microbiome perspective, just from other perspectives. But from it, I'll go back to I'll come at it from the microbiome perspective and kind of speak to it from what we know to be good. So I would say, and it is really hard because it really depends. Again, it goes to like how much, for what, and the truth is that we're moving towards a place where we're getting to such large data sets that the field so far has, I don't want to say has been guessing, but has been highly correlative. And now we're coming to the place with these like huge data sets that are in some cases going to be 5, 10, soon 15 years old. And the answer to the question will be for you or for me, right? And it will be, and the answer for you may not be that it's right for me, or which ones, or red meat versus poultry, So I'd say that there's a lot of nuance to that, but I would, just to start from what we know about the microbiome, we know how important plants are. We know that from the Human American Gut Project, and like all the work out of UCSD and that big citizen science project that followed the Human Microbiome Project, that 30, like, I think they looked at this interesting stat that came out of it that was like 30 different types of plants every week led to like a more diverse microbiome. And I think that's important just because people who eat mostly plant-based, you end up in a lot of the same loops.
Superfoods.
Yeah, superfoods,
right?
or the loops,
Yeah.
But actually the diversity was something that was really, I think, really interesting to think about. And it's all at different levels, right? Like, sure, eat the same plants every day if you're not eating a Big Mac, right? Every day. So, of course, it depends on food access. It depends on a lot of different things. But for the most part, I thought that was really fascinating, that the different types of plants and, of course, the different, the variety of them. And then, of course, what you were referring to earlier in terms of the different types of fibers, and then all the phytonutrients and the phytochemicals that your microbes then use from those, and how important fiber is and how important those prebiotic compounds can be to the proliferation of good bacteria, to the flourishing of this rainforest that you're trying to keep dense and healthy, and of course to the function that you want them to be able to have, so that they have enough of these compounds that they can continue to make all this really good stuff for your body. And so I think that plant-based, or primarily plant-based, continues to be, to me, really important. But then you have to say, okay, well, what about like omega-3s? Because those are really important for the microbiome too. And you can look to certainly olive oil, and you can look to avocados, and you can look to certain foods, plant-based foods, absolutely. But I think that there's argumentation for sardines and some of the smaller anchovies and some of the smaller fish that don't accumulate metals and can be a little bit more responsibly gotten.
Yeah.
But it's hard to, sometimes you say these things and you have to remember that not all these suggestions are sustainable. So that's beyond the human health.
Yeah. So with the consumption of meat, especially, CAFO or industrial meat, where they're using antibiotics.
Exactly. That's where I was heading, which is like, it's so, and then when you come back to meat, which is, again, I'm trying not to be like circuitous, but it is a really hard, it's a hard topic. I know people who, as I said, I think in the future, we're going to know a lot more about like whether it's right for you and maybe not for me. And I'm actually not somebody who's obsessed with personalization. I think there's a lot of interesting, things that are going to come in the future, but I don't have like this obsession that everything in the future will be personalized. To your point about the Human Genome Project, sometimes we're not that exciting, but I do think that, and the debate has gone back and forth even within our company and even in science. A lot of people debate this all the time. Meat is something for me that, like, when you, yes, there's the TMAO discussion, and there's certain aspects. In fact, as I said, some of the research that we're currently doing that was not conclusive yet, but hopefully next time we do this, I can speak more conclusively from a microbiome perspective. But I will say that I think that if constituting a small percentage, if you are going to eat it, but I also think that the meat, particularly in this country, is not, and I'm coming to you after caring for my husband of 2 days off of the worst food poisoning he's ever had in his entire life, off of a turkey meatball from a very quote unquote healthy and expensive healthy place. So I do think that it's not so much looking at meat in isolation as like what you were saying, which is that, depending on where you're getting it, particularly in this country, between antibiotics and all of the other and just like the soil, what they're eating, the water sources, the processing, there's so many things that, it's nice for some of the people that are in maybe more of the wellness world who like go off and like kill their own meat. And certainly there's ways to do it that absolutely would be more supportive and of course more sustainable. But it is hard for someone who likes myself to think more about like less even about some of the science and actual chemistry that you're talking about, like the compounds and inflammation and things like that, which I think you could argue with anyone for hours about. There's just the aspect of like, it's just not sustainable.
Yeah. But it doesn't come down to, for example, when you feed a particular strain of bacteria fiber, it produces butyrate. But if you feed some other strain of bacteria, animal protein or meat, it produces hydrogen sulfide or something.
I see what you're saying. The fermentative aspects of meat, I to be honest with you, I'm not as familiar with. But to qualify, I don't know. I don't think the way that like the amino acids would be more what would be probably at that point of digestion be interacting. So if it's okay with you, I'll get back to you for the show notes for that, because I need to think a little bit about that. But they wouldn't be like, it's not like, oh, meat as a prebiotic makes this and therefore it's bad.
Right.
It would be, I'll get back to you on the meat, the meat conversation.
What about sugar?
Yeah, I mean, sugar, I mean, the other interesting thing about microbes is that they start kind of what you're saying about maybe attracting who you love, or having some sort of like evolutionary predisposition for you to like procreate with somebody through your armpit bacteria. Microbes are what you feed them and what you give them on a regular basis has a lot to do with what you continue to crave. And so they definitely start to, because they start to self-select then and become the dominant voice in what you eat. And so sugar is a big one that actually can be very perturbing, especially like refined and processed sugars, particularly ones that are not necessarily being accompanied by any kind of from a fiber perspective. That's what you find more in like fruits, for example. But yes, sugar is a big one. Sugar and processed foods in general. I mean, just refined sugars are absolutely like, I mean, certain microbes like them, but not the microbes that you want to like them are liking them, and then they'll keep signaling to crave them.
Right. So off diet for a second. I mean, the last few years have been dominated by fear and anxiety and stress.
Yes.
And the relationship between stress and your endocrine system and the secretion of certain kinds of hormones, et cetera. Yes. Can you pull on that thread a tiny bit in terms of the relationship between stress and gut balance?
Yeah, of course, absolutely. I mean, the gut-brain axis, which is this kind of bidirectional highway that exists between the gut and the brain, which is primarily primarily through the vagus nerve, and is kind of where most of the communication happens. Although they're finding kind of new signaling and all kinds of connectivity all the time, and starting to look at more and more ways. But for the most part, it is bidirectional, which is important because it's not so much this like, I think one of the ways that it gets like a little sensationalized is the gut's telling your brain what to do, but there's a very like two-way walkie-talkie here. And so stress is one of the areas that they have found, actually your microbiome, even prior to like where gut-brain axis research is today, I mean, your gut really responds to stress. I think there's like some really interesting studies that were done a while ago of like the microbiome in a crowded elevator. And if you're somebody who's like more claustrophobic, and you could see like the impact of a stressful situation on the microbiome. And that was before really understanding like what kind of signaling is happening, and so I think you absolutely, I mean, stress is a really big one, which is one of the ones when I was going to talk about, when you asked me the question of like, what are all the things that can hurt it, but stress is a really big one. And now what we're starting to understand, and actually one of our scientists who's on our board, and probably one of the best scientists working in the field of gut brain today, Dr. Sarkis Mazmanian at Caltech, who's right here, is working on really understanding how microbes and the metabolites that they produce suppress or increase like our stress response. A lot of his work is in autism as well. So really interesting to look at like neuropsychiatric conditions or diseases where, of course you see these things like really exaggerated, and what that looks like, and what that metabolic signature looks like, and what's happening. But he was one of the first to identify in mice, and has now just published in first human study, one of the metabolites that's responsible for things like stress response and social behavior, which is really interesting. And so your microbiome very much responds to stress. It is one of the greatest perturbations, and I think that's probably why you see such dysbiosis in people with like IBS, IBD, Crohn's, things. And one of the challenges with all of these, even with autism, and why it's so important that the science moves forward to understand like how this is working mechanistically, is that so many of the areas there's so much correlation between GI distress and some of these neuropsychiatric conditions, and you get a bit into the chicken and egg there too, because when you ameliorate the GI distress, you also ameliorate these feelings of anxiety and stress. And so what's hard to know is that did the GI condition, and you see that sometimes in autism, because you have kids who really suffer and have a lot of GI distress. And then you sometimes, there's a lot of research, early research, some of it a little bit sensationalized, on probiotics for children with autism. And so you would see that like just by like increasing their bowel frequency, or helping them go to the bathroom more easily, or reducing bloating and some of the cramping, some of the things that can come from very specific strains, their symptoms would get better. So of course people say, probiotics cure autism. And so I think there's a lot more to unpack there, but people like Dr. Mazmanian and others are now really like, there's a lot of clarity about the mechanism, what's working, particularly in mice. There's a lot of evidence, but as we know, now needs to be brought into humans. But we do know that the microbiome is incredibly responsive to stress, and it's a really big one that we see obviously exacerbate many conditions and quality of life for people too.
Yeah, I mean, I think a lot of these chicken and the egg issues come down just to this notion that all of these systems are connected.
Yeah, absolutely.
And I mean, I think about sleep and gut dysbiosis, for example, and you can have trouble sleeping because you have sleep apnea, or a whole myriad reasons why you might have trouble sleeping, and that will raise your cortisol levels.
Exactly.
And high chronic cortisol levels will have an impact on your gut, which is just detrimental.
Yes.
At the same time, you might have a gut in dysbiosis, and that's prohibiting you from getting good sleep, which has all sorts of downstream impacts.
Correct, yes.
So it's so it is
and
really
microbes
hard.
have their own circadian rhythm.
Is that right?
Yep. And they need rest.
Oh, that's right.
They need rest too. They see this the way that one of the most interesting things I've seen them study in this world is that you'll see dysbiosis in not just other disruptions, but they'll study like shift workers, and not only shift workers, but they'll see that like the dysbiosis is related to, of course, the disruption of the circadian rhythms, not just of the human, but also microbes have their own circadian rhythm that's really important, and they need to rest and do a lot of really important things at night, so that they can, of course, do their important functions during the day. And so when you see that get out of sync, and you see that a lot of people think, and there's some hypotheses around jet lag, and whether or not what's kind of happening between microbiome and the human, but also in shift workers, it wasn't even just in the shift workers themselves that they saw disruptions in dysbiosis, but they also saw it in something called secondhand shift work, which is the nuclear family of someone who's a shift worker. And the impact of their shifts, even without like huge disruptions, but you would see it like impacting the nuclear family or whomever you're living with as well. And then their some of the biomarkers that they could study to see what those disruptions did too, which is fascinating. It just shows you how wrong we've gotten everything.
Yeah, I mean, you could even think about light, for example, that in humans sets the circadian rhythm. There's these little neurons, melanopses, I think, in the bottom of the retina that sense light at a certain time in the morning. That's why it's always important to try to go outside and get light in the morning, which sets your circadian rhythm, which essentially regulates the secretion of cortisol, melatonin at certain times in the day. And if you're not getting that light, or, for example, if you're getting blue light from your phone or from your computer screen, binging Netflix late into the night, well, that's going to screw up your circadian rhythm. And by extension, your endocrine system and the release of certain hormones at certain times of the day, which could then, of course, have downstream impacts on your gut. So it's all of this stuff is just
and really interesting things that we see now being correlated with like metabolic disruptions, right? So if the gut and brain connection is a big part of like signaling satiety, like when you're satiated, right, when you should stop eating, you can just see how those little disruptions, and then that signal is interrupted, and it's like, wait, did you say we're full? Can't hear, I can't, bad signal, and then of course you start to see that then over, the increase of body weight and or overconsumption of food, and so you just start to see how these things impact each other. Or, for example, something as simple as like the neurotransmitters that impact motility. Right. So like a lot of people also sensationalize that stat that 90% of the serotonin in your body is produced in the gut. True, but not all for regulation of your mood. Also, it's really important in just like making sure you poop well, because that stimulates the motility of your bowel. And so, but you just start to see how these things, when these signals and all this, this chemistry is obviously disrupted, and then you have, a big part of the population that like is basically clinically constipated. You have right now 14% of American children are clinically diagnosed as constipated.
Wow.
Which is incredible,
Yeah, so I was actually going to ask you about that as one of these kind of mythologies of the microbiome, because you're so good at unveiling them. So yeah, the serotonin one, for example. And serotonin is interesting because it's actually the precursor to melatonin. So you really need it for a whole variety of reasons. It's not just this feel-good hormone. But yeah, you hear that statistic thrown around a lot. 90% of your serotonin is made in your gut. Now, is that true? And is that cross the blood-brain barrier? So if you have a happy gut, will you have like a happy mood, or is that kind of a stretch?
I think it's again. I think that there's there's no shortage of people on Instagram that would agree with you.
Okay.
I don't know if it's that one-to-one, nor do I think there's necessarily evidence to support that, given how many other factors impact it. I would say it's a really important contributing factor. Microbes, as far as we know today, do not cross the blood-brain barrier, although we do have a really interesting study with the NIH to determine if there are any microbes in the brain, which is like a really cool thing, because to date they've been known to, of course, be sterile. But of course, those are the compounds and obviously some of the metabolites that do cross the blood-brain barrier, and a really important part of that, like that crosstalk. I think the stat is, I have to look up the latest like citation. There's always some newer number or percentage, and I'm always hesitant just because the mainstream kind of people. And by the way, so you see this in science, like you'll still see scientific papers today that use the 10 to 1
all the time.
Which is incredible. And really interesting. But in terms of the percentage, it's a very high percentage. I don't know if that's the exact percentage. It would be misleading to think it was just for the regulation of mood and anxiety, and all the things that you kind of think of serotonin as, and just like precursors to like things like melatonin, because it does have just a very mechanical utility, which is that it's an important neurotransmitter that does very important things like the elimination of waste too. Which I think people just don't think about a lot.
So let's say that, you're eating fiber and you've got a good, healthy plethora and diversity of bacteria in your gut and everything's cranking well, that can really influence a whole variety of axes of health. And here's another statistic that's thrown around a lot, and maybe you can unwind it, that 70 to 80% of our immune system is in our gut. So what is the relationship between a healthy gut and a healthy immune system?
Yeah, I mean, it kind of starts, there's a lot of crosstalk between like a gut and immune cells. And the percentage is probably pretty close. I think that has a lot to do with what happens in like early childhood development and where our immune system and how our gut kind of develop and co-evolve. But if you think about it, there's a tremendous amount of information that's coming in to the gut on a daily. Obviously in the form of anything ingested that we need to live. And early on in like in early childhood development, the gut and the immune system are they're kind of developing at the same time. So, you have the early, infant microbiome that is incredibly malleable, and that's developing in the GI tract of the baby. And all of that like early training is happening in the GI tract and with those microbes. That's training your immune system, hopefully in a healthy way. Of course, what we know now is that the correlates, and even now kind of pretty clear, more than correlative, but pretty clear direct connections between everything from allergies to autoimmune conditions to even certain areas like autism, that we're starting to see that there's, during these developmental windows, when our microbiome is forming and solidifying into what they call a steady state, which is, scientists say, somewhere between 3 and 5 years old. There are these windows where the GI tract is developing along with microbes and your immune system. And this is where like the friend or foe, learning like what's good, what's bad. Do I have a response to that? Do I not have a response to that? And of course, there's so many factors that influence from the minute you're born, even before you're born, now that we know, that maternal, speaking of which, all the way through, those first formative years is really where there's this kind of like co-development period where there, of course, your GI tract is developing and starting to, of course, starts with breast milk and certain carbohydrates and the nutrients that are coming from either breast milk or formula. But then, of course, a lot of information that's coming from the outside world. And really during that time is when the immune system is learning, right? And that's the training wheel period. And then by the time you're at a steady state microbiome, you know, a lot of things like either whether food allergies have either developed or not developed, or the the foundation for whether they will develop has certainly been laid in some way, or whether or not your immune system will kind of misfire, or there'll be other kind of good or bad outcomes from that period, depending on what information and what kind of help shaped it environmentally. And so that if you think about just the way it develops at a very early stage, you start to understand like how important that crosstalk is, and why the immune system is basically based there, and why those formative years are so impactful to kind of the way that your immune system then functions really for life. And so they're incredibly and inextricably intertwined. I think the stats that I've seen are that number of 70 to 80% it's about right. Certainly when you think about like the way it co-develops and how it's kind of stabilized and formed. But obviously there's so many other factors that impact how it all functions. And we talked about like permeability. We talked about like the microbes are such a big part, just to show like one mechanical example of such a big part of the integrity of that gut wall. So like a really beautiful circular example is like microbes metabolize certain compounds and make short chain fatty acid called butyrate. Butyrate is like the lead energy source for the colonocytes in your gut. And that, of course, is what maintains the integrity of it. And when that integrity is broken, you get things like leaky gut, and you get poorly or misinformed triggers of the immune system. And so you can just see how circular, and then of course there's many other examples.
That's the
like
coolest thing. When I read that, I think I might have read that on your website because you guys have so much good information on seed.com. Because I always associate energy production with either glucose or free fatty acids or ketones in some cases, if you're in ketosis. But you don't really associate the energy production from butyrate or from metabolites. And I'm like, that is coevolution in a nutshell right there. 100%. It's amazing. Yeah,
exactly. And also just going back to the genome versus microbiome and agency, and there are foods and these plant fibers and phytonutrients and phytochemicals that allow your microbes to make more of that stuff, and so of that good stuff. And so you can also just see how you play a role in the choices that you make every day, both consciously and some that are not, particularly environmentally, that we don't always make for ourselves, of areas where it can be conscious and areas where we have a lot of agency in that circularity.
Yeah, I was also reading about the impact of metabolites, I think butyrate, on T cell differentiation, specifically around T regulatory cells. This gets a little down the rabbit hole here.
Yes, it does.
But essentially these immune cells that are T cells, which are central to your immune system, T regulatory cells, to the degree that I understand it, help to sort of suppress overreaction of the immune system. So we heard a lot about like the cytokine storms in COVID and whatnot. And I believe that there actually are some preliminary studies that have shown that the more severe contraction of COVID has some correlation with depleted microbiome.
That came out recently. We talk so much about Tregs at Seed that we have a sweatshirt, a T regulation sweatshirt that we made for one of our scientists, Dr. Aza Gadir, who was our director of R&D. She's now one of our scientific advisors. Her seminal kind of culmination of her work at Harvard before she had joined us was looking at Tregs and their development of childhood allergies. And she had a cover of Nature paper, which if you don't know is like very fancy in science. Yeah, it's like cover. That's like cover of American Vogue. Yeah. And so she American Vogue, and she not Hong Kong Vogue, and she. And her paper was about Tregs and the development of allergies, and actually that there's this switch that they found that actually could be flipped for kids with, and that worked for all allergic responses to food except for shellfish. So, and it had to do with, of course, Tregs and the specific pathway that they had discovered, and that would be using a very specific microbe to flip that switch, which is really fascinating. Wow. So there's a lot, there's a lot there. It's not a big focus of our work at Seed, outside of a little bit of some of the gut-brain work that we're working on with Dr. Mesmanian, but it's a whole other area that I think we're going to start to know so much more about. And the Treg stuff for COVID came out, but there's a lot of, really interesting research about Tregs, allergies, and of course the development of other like autoimmune conditions too, and the role of the microbiome in that.
Mm-hmm. So there's a number of studies out there, kind of switching gears a little bit to metabolism and the gut, particularly in vivo mice studies where they'll take the gut flora of a fat mouse and put it in a skinny mouse and feed them the same diet, and the skin or they don't change the diet of the skinny mouse, but the skinny mouse gets fat,
and the fat mouse gets lean.
Yes. So what's going on there? Is that upregulating insulin sensitivity, or what's happening?
Well, actually, there's two studies that happened. That one was in 2006. I think I believe it was from Jeff Gordon's lab, and then there's another one that I believe was a few years later, that also demonstrated that you could make the mouses fur much more lush and shiny.
Yes.
Which just really shows you how science is so. Still a slave to the things that we value as a society, because the field didn't really pop until you could show that people could look younger and lose weight, which is honestly, how many areas, particularly you see this with longevity now, and you see this with other areas, like people don't pay attention to until you can show that you could lose weight and look better, younger in this case.
Yeah. Or live to 150.
And I would say I don't think from that mouse study that they were able to pinpoint like this is specifically like the mechanism, but the reason it was like that shot heard around the scientific world and why it blew up the science internet is because it was one of the first ways that you could actually demonstrate how powerful the microbiome was from a metabolic, perspective, in terms of like metabolism, weight management, and the fact that these transplantations, which are being used for a number of things today. I think we believe the future will have a lot more specificity to it, but of course, like as an interim solution, I think a lot of people are finding success with what they call FMTs, which are these fecal microbiota transplants,
Right.
where you do take a healthy microbiome and transplant it into someone who has a specific condition endpoint, or something that's resulting from what someone believes will be dysbiosis. And there's certain areas where like with certain skin conditions, like I believe like atopic dermatitis, psoriasis, like they've definitely shown, even taking that same methodology, to have an impact. And there's other areas too that FMT is being used. I think, again, we just think it's not as precise as where we're heading. We'll be able to isolate the microbe or the metabolite and be able to administer it directly. But what that did tell us was that the role that microbes play in metabolism, and then of course that has opened. And I think what we're going to start to understand now, and that was why I was alluding to those like really big data sets. I think there's been a few microbes that have been identified that people have tried to kind of correlate with weight loss and with metabolic health, and some with better or worse, some with better data, some with maybe just needs to learn more data. But I think what we're excited about in the future is these huge data sets where you've looked at like large populations over long periods of time, and you've started to understand like these very specific markers or metabolic signatures, and where you can start to really understand like what's actually happening, what microbe could be used to do this specific thing, and who would be a responder and who would not be a responder. And I think that that's coming. It's just a few years away.
Yeah, there's a lot of variables in figuring that out.
Yeah, because when you do a full transplantation like they did with those mice, it's like it can tell you like, holy shit, we got to look at this and understand it. But what you don't then end up knowing is like, well, which you'd have to take out and isolate which microbe did what,
Right.
Why did it do that. But of course, what it did unpack and like blow up for the scientific world was, oh wow, like we now need to focus here, because that tells us a lot about the myriad of functions that are happening from a microbiome perspective, and like that we need to really start unpacking this with more specificity.
There does seem to be some good data supporting fecal transplants for C. diff, right, in particular.
Yes, some, yeah. But not entirely curative, but yes, a lot of people have had, and that's always the case, like right with things that are considered unmet medical needs, or like people are just dying, like you something that's working is better than like
right.
the thing that's going to work across everything. But yes, there have been, although a lot of those trials have also failed to proceed and and get regulatory approval too.
Yeah, it does seem that there are clues that there's some crosstalk between the genomics of our microbiome and our genomics and our human genomics.
And another factor, and even other factors. Yeah, absolutely. I think that what we refer to as like these multi-omics data sets, which is like everything from capturing dietary data to genomics to metabolomics to microbiomics, and then using bioinformatics to start looking at these big sets and saying like, oh wow, like this person would be a responder, this person, you start to see these correlations, and that has to do with absolutely some of these genomic variations that can happen at the microbial level or at the human level.
Okay, one more question, and then I want to talk a little bit about when the microbiome actually gets seeded.
Oh, sure.
Because this is also a personal topic for
Yes.
you. But as it pertains to cognition and I suppose neurodegenerative disease. I've seen some folks posit that that metabolites can reduce the deposition of like beta amyloid proteins in the brain, for example. Do you see a direct connection between what's happening with the production of SCFAs and metabolites and dementia?
And dementia, all the, I mean, it's another area that's really the other I guess dimension to even bring into that conversation is not just like gut-brain axis and the metabolites from the brain and the role that they might be playing in the advancement or acceleration or prevention of the onset of neuropsychiatric conditions, but also like really interesting to understand that like a lot of some oral microbiome research is moving there too, such that they're starting to understand like the role that the oral microbiome, and if you think about it geographically, right, like it's very close, maybe playing also in the onset of those conditions. And that's another area too that we have a research track that we're going to be announcing soon that goes a little bit more into both gut brain, and then obviously our oral microbiome stuff is coming up soon, which is with the Forsyth Institute at Harvard, which we're incredibly excited about, and that's looking a little bit more locally at the oral microbiome, but they have done a tremendous amount of work starting to understand not just gut brain, but how the microbes in our mouth play a role in everything from respiratory health, cardiovascular health, and then also, of course, neuropsychiatric conditions, which is really fascinating. So it may not just be gut as well that's impacting the, as I said, acceleration and or prevention of onset of some of those conditions.
Yeah. Well, this is the exciting part about being in this work, right? Because every day something new is happening.
100%. Yes. While we're sitting here, I'm sure like one of my stats is outdated.
Okay. So as it pertains to the development of a healthy microbiome, when does the microbiome actually get seeded? Do we know?
Oh, this is a wild controversy in science. I would say until a few years ago, most scientists would say something like the womb is sterile and you are first seeded with microbes at birth. And seeding is simply, which is where our name comes from as a company, seeding is the process by which, biological process by which an infant is first exposed to microbes. So important to note that the seeding definition does not say at birth, although that used to be the like way most scientists or clinicians would answer the question. What we're now understanding is that there may be some shaping that happens. What we don't entirely know about microbes in the womb, but we do know that the metabolites are crossing from an amniotic perspective are coming from or into the fluid, and we don't know, and what a lot of people are starting to look at is how that is actually starting to shape the infant's microbiome even prior to birth. And so there are a few things that are like correlative that have been kind of like established. There's really interesting early research around the role of the mother's nutrition and the development of the child's microbiome, regardless of environment and other things that happen. So like what the mother ate and their microbiome, and then the relationship between the development of and what you start to see at even as late as like 18 months in terms of like BMI and obesity profiles from the nutrition of the mother. And that they believe that that's coming from a microbiome perspective, like informing both between the metabolic exchange that happens or the metabolite exchange that happens prenatally. It impacts nutritional profile of breast milk, but it also impacts the early seeding because, of course, the mother's microbiome and the metabolites that are coming from that are a part of that seeding process. And then, of course, having an impact on what that child's microbial signature looks like after. And then, of course, there's like nuance like post-birth, which is when I don't think anyone's disputed that the mother load, so to speak, comes at birth. And that's of actual microbes. And that, in a vaginal birth, is being seeded by vaginal microbes, fecal microbes, and then the mother's skin microbes. Those are like the first three like real exposures. And then, of course, wherever the environment where the infant's born. But those are probably like the first three constituents. In a C-section birth, you'll see the early microbial signature of a baby look more like the skin. And that's just simply because they're not passing through the vaginal canal and they're not exposed to any of the fecal microbes, which, whether or not you poop during birth, there's still microbes that are
Around.
translocating in that general area, which a lot of people and my co-founders kind of talked about this before. There's some belief that there might have been like an evolutionary reason for that, which is kind of interesting.
Yeah.
Especially when you look at a lot of animal species that eat their mother's poop, or where the poop is a part of early diet, and or early like more conscious exposure in like certain mammals, and actually even in like some insects and stuff, which is kind of interesting. So even like baby koalas eat their mom's poop as like a primary nutritional source. It's called PAP, which is kind of really kind of interesting. So just to show you evolutionarily, like there's a lot of rationale for the proximity.
Yeah. And in C-section birth, is swabbing becoming more common practice? And what would you
It's a great
say
question.
about that?
Gloria Domingos-Bello, who's an incredible scientist, she's now at Rutgers. She was at NYU for a really long time. She's out of New York, now New Jersey. She is probably the like foremost scientist in the swabbing world. She's had a lot of regulatory pushback on swabbing.
Really?
Yep, really fascinating. Yeah, the FDA doesn't love it. I can't really understand the susceptibility to infection. I'm like, look, 5 minutes before it was going to come out and see it and be exposed to it anyway. I'm like, I don't really see the difference, but there's been some pushback on it. Just feeling like you don't really know whether there could be some infection. Partially from the hypothesis that if the baby didn't come out vaginally, perhaps that was also, there's a reason that there might be something microbially that's going on that you wouldn't want to necessarily transfer. But for the most part, I think, I also just know a lot of doulas and midwives who just kind of do it anyway. But there's early research to suggest, honestly, good and some, I would say not like questionable, just things that need like further research as to whether or not that would become standard. But I do think the C-section versus vaginal does get a little bit conflated because or contrived, because there are other factors that go into, some people are like, oh, a C-section, like I'm fucked for life. And I'm like, well, it's not exactly like that, because what they do see, and I think Rob Knight talks about this in his TED Talk or has published about this. He's a really well-known microbiome scientist from UCSD, which is, in the presence of breastfeeding and in the absence of antibiotics in the first 18 months, so being breastfed, but no courses of antibiotics, you do start to see a convergence of the microbiomes, depending on, environmental, there's other environmental factors and other things that kind of go into that.
Vaginally versus C-section.
Yeah. So, so I think that there are, to our point that we've made a few times while we've been talking, like some areas of where the malleability of the microbiome really serves you in those early months, those formative months where like the blueprint is forming, because there are choices you can make there. Being very cognizant that breastfeeding isn't an option for all mothers, both environmentally as well as nutritionally, that can, impact whether or not, you kind of can start on that kind of towards a steady state microbiome from like a place that's kind of on par with a more traditional vaginal birth.
Mm-hmm. And maybe talk about the breast milk a little bit, because as I understand it, there's a significant component of it that's actually not digestible.
Yeah. HMOs, not to be confused with like horrible places to try and submit your medical bills, are human milk oligosaccharides. And those, about a third of the carbohydrates in breast milk are not digestible by the human part of the baby. They're really just a substrate, kind of like the fertilizer for microbes to flourish and to grow. And so there's a beautiful evolutionary story between breast milk and babies, because everything from the microbes that evolved to live on the nipple, which is crazy, microbes that are or the way that the saliva in a baby's mouth, there's like bidirectional microbial activity between the baby's mouth and the nipple that you can't see, which is incredible. Some of which, for example, were selected because they're microbes that are good for helping the baby digest lactose, as an example, just to show you like how evolution is like insane. And so of course that's like a first interaction, and then of course the bacterial. There's microbes in breast milk, so there's a breast milk microbiome, and then there's the nutritional profile that includes those like the sugars, the HMOs that I just described, and those are very much, in fact, now where a lot of science is going is looking at how you could synthesize many of those microbes to include in formula, so that babies who have to have formula, you can add certain microbes, and you could also be adding as much of the full spectrum of HMOs that are present in breast milk as possible to the formula, so that you can start to replicate and get as close as possible to some of the really important exchanges that are happening during that window where the microbiome is developing and where breast milk can be such an important accelerant of that, of the health of a healthy development.
And are there ways, essentially, if you're not able to breastfeed, and are there kind of pediatric microbiome supplements or products?
I mean, it's early in the field. There are. It's just early in the field, so
yeah.
There are a lot of formulas now include certain probiotics, and they'll say and certain HMOs. I mean, there's definitely a lot of formulas, particularly the number of the ones that are maybe a little bit more scientifically or evidence-based that come out of Europe, that absolutely are looking at the nutritional profile of breast milk as it relates to, so looking at things like omega-3, looking at certain compounds as it relates to healthy nourishment of the microbiome. They're looking at which microbes they could use to supplement into, so probiotics that are in the formula. And then they're looking at, as I said, just like how they could synthesize and replicate. There's a huge spectrum of HMOs that are in breast milk. And so a lot of them, you'll start to see on marketing, like on the outside of cartons, like 24 HMOs or 16 HMOs, which is like the new way of looking at formula. But I would say a lot of that research is a little bit early, but that is a really promising part of the field for sure, in terms of formulas that will start to get closer to, or will be developed actually from the dimension of not just like the human nutritional profile needs, but actually from like the incredibly important like microbiome needs during that window of development. And I think we're doing a lot of really interesting work in that area. And you're going to start to see it'll just be commonplace and table stakes that every formula will have probiotics and probably some aspect of HMOs.
So humans aren't the only beings that have microbiome, right?
Correct. Almost all living organisms have a microbiome. And
I know that you guys are doing specific work as it relates to honeybees, right?
Yes, and coral reefs.
What happens, I guess, first if we don't have any honeybees? Because what I've read recently is that at least in America, I think we're down to like 89%. Or I've seen a whole bunch of statistics that seem alarming. And without pollinators, like I think something like 80% of the crops just disappear. So
and animal species too, right? Because the ecosystems,
So what are the primary causes for this diminishing population of honeybees?
I mean, there's a number of causes. I mean, the one that we look at the most, we look at two areas, honeybee, colony collapse disorder, and an American full-brood disease, which is actually interesting, lesser known, but important because it's really where the larvae die. And so and you just see like huge amounts of die-off. And look, a lot of it is like probably won't surprise you just based on like things like, our work primarily looks at pesticide use, particularly the neonicotinoid pesticides, which is what we focus on because implied in their name, they have nicotine. You can see in certain studies like bees that will literally not go to nectar and they will go to the pesticide first because of how addictive it is. And so it's incredible, you just think about, first of all, how sick someone was like once sitting in a lab somewhere and they're like, I got it.
The bliss point for
I got
bees.
it. It's going to be Marlboro for honeybees. And so I think I think that it has to do a lot with a lot of our work has to do more with like the pesticide use, just because of the direct impact it has on the gut microbiome of the honeybees, and then how it impacts their immune resistance. And so it brings their immune systems like way down, unable to, effectively, fire the right responses, and have any kind of, and also impacts, just like antibiotics and all these other antimicrobials, has a huge impact on dysbiosis, and then of course they die off. And so we created this biopatty, and it can be administered as a spray now too, of very specific microbes that actually increase the immune tolerance of bees against these pesticides. So not a perfect sustainable solution. It doesn't change the systemic issue, but certainly allows them to sustain themselves, which I think is something that a lot of people working in climate change, like we were talking about short-term versus long-term thinking, sometimes you do need to implement some of these shorter-term solutions so that you can buy the time to get to the more systemic and longer-term solutions.
So these are like literally look like
they look like pancakes. They look like pancakes. They actually look like pancakes. I can send you a picture of one. They literally look like, they're literally made. One of the things we have found, in what we do, and you probably see this a lot in like anything you see in health or wellness, is that human behavior is such a big part of it, whether or not some of these things are effective. So we didn't come up with the idea of putting a pancake in the hive. You just have to ask beekeepers and the people who own these farms, and who obviously can be the administrators, of like, what would be the easiest way for you to administer something. And that is already how they feed a lot of hives. And so we first demonstrated it in the patties, and then we made it even easier, which is literally just go around the hives and spray it.
Right. So I'm gonna ask a really dumb question.
Sure.
So wouldn't the farmers that apply the pesticides need pollinators? So why are they killing off their bees?
Yeah, it's a great question. I mean, that's a really good question. A lot of these, first of all, a lot of these bees are not bees that are being used necessarily for the farms. So that's the first. Okay. That's the first piece. And a lot of those pesticides are not being used to kill bees. They're being used to kill all the other animals, and they're not like actually like trying to kill bees as much as the exposure in all of these ecosystems is what's killing them. If that makes sense.
Yeah. And do you concentrate any of your work on, for example, glyphosate gets talked about a lot, but on chemical herbicides that are essentially chelating essential minerals in the soil and contributing to soil degradation and erosion and et cetera?
Our work in soil is more there's a couple areas that are our work in soil will be looking a bit more at, although it's related, but for us, how you can use microbes to increase nutrient density of food. So even, I guess kind of what I was saying in the short term versus long term, which is, what are the microbes that could be administered to create the conditions, despite dysbiosis of the soil, to improve nutrient density of food, which is of course a huge problem. That's just our work. There's very, very interesting work happening across soil microbiome everywhere, but that just happens to be one of our areas of focus. And then the second, which is not the way that you describe soil, but if you took the same sentence and said the things we're exposed to in our homes or the built environment, that is a big area that's coming up in our work, which is starting to look at the compounds, including the ones that are quote unquote natural, and what they do to compromise our skin microbiome, our gut microbiome, what it means to be wearing clothes all day that with very specific surfactants and chemicals that were used to clean them, and what that's meaning for our health, and what are the new materials and compounds that will be far more nurturing, and starting to think about surfactants and the compounds that we more live with, as a part of our kind of proactive aspects of health.
I know those are more like compounds that are in kind of daily cleaning products and et cetera.
Yeah, absolutely. Which there's incredible research that's coming out, as well as that has already come out on the impact. I mean, there was a paper, I think 2-3 years ago, just on like the correlation between childhood obesity and home cleaning products, just to understand like how, look, you're in such close proximity, and you're already in the built environment, which means you're already in a more a microbially, despite what people think of the New York City subway, it is not as microbially diverse, and you're already not exposed to the endotoxins that like all Amish children who have no allergies are. You're not exposed to so many things that we used to be, but more importantly, you're not only not exposed to those things, but you're now exposed to so many chemicals and compounds that are disrupting to everything from your skin barrier to your gut barrier to all these things that, of course, even, again, as I said, even some of the more natural ones that we're kind of just starting to understand the impact on our health from.
And I suppose that plays into this hygiene hypothesis.
Yeah, and the missing microbes hypothesis, which is kind of a part of that, which is like hygiene hypothesis is essentially this notion that we've over-sanitized ourselves to sickness. Yeah. And then just to be like, it's kind of reductive. And then missing microbes is this notion that because of that, and on top of the overuse of antibiotics, and on top of living in the built environment, and on top of the decline of our both nutrient density and then just quality of diet, everything from just the misproportioned ratio of omega-6s to omega-3s, and the lack of fiber. Missing microbes is a little bit more akin to this idea of, which is off of hygiene hypothesis, but a little bit broader, that the rise of all of these non-communicable diseases and autoimmune conditions is a result of basically what I would refer to as the climate change of our insides, which is, if you think you think it's terrifying that we've lost 50, 60, 70% of the North American bird species. Wait till you see how many species we've lost in here. Yeah. And how that, our bodies are analogs for what's happening out in nature and the environment, and that missing microbes posits that the decline of speciation, and not just diversity, but the richness profile, so how much and how abundant and how many different types, is really highly correlated with the rise of all of these misfirings, like asthma, autoimmune conditions, allergies, because we're just simply like, there's no training, there's no proper training ground, if that makes sense, from what we're talking about for early development.
Is there a spiritual component to your work?
I think I'm a very spiritual person, and I think science is a beautifully incredible, and I think the microbiome is an even more, I think, greater expression of, if you define spiritual as the going inwards or the knowing of oneself, which I think is kind of one of the definitions. The entire, forget industry and forget people's feelings about big pharma and, when it gets commercialized, but I think as a discipline, and I think you can look back in history to see how even astrology and astronomy were kind of reconciled at the same time, and how medicine and, all these people were not dividing themselves the way we are divided today. And I think, and by the way, some of the greatest artists were the greatest scientists, and scientists greatest artists. And so in so many ways, I see them as very, I see my work now as like more spiritual than anything else. And so I think science, it's a discipline of inquiry, which is actually in some ways less biased than like religion, and less biased than saying I am this or I am that. It just says that I'm committed to asking questions and wanting to know more, which I think is like a beautiful. And then I think the microbiome, in Star Wars, of course, if the midichlorian, a lot of people think that those might be microbes, or that George Lucas was kind of prophetic in some ways about, what microbes could be doing. But, I think if the microbiome's a good teacher in anything, it's that you could be looking in the wrong place for a really long time and basing your entire worldview and truth on it, just because you simply didn't have a way to see it. And so I think we see that in everything we do. What I am leery of is it is kind of what I said. I think we talked about this a little bit on our first podcast, which is it's the hypocrisy of confirmation bias from the very same people or disciplines that are grounded in this notion of open-mindedness, and seeing everything while also being very anti
Yeah.
things that they both fundamentally don't really actually take the time to understand sometimes. But just because it doesn't fit within the way the world has said you could be Buddhist and believe in vaccines, right?
Yeah.
Like, or the way that people like tie these dots together inextricably in a discipline where you're concurrently talking about how open-minded and how the world needs to be one and all of these things, and not realizing the hypocrisy of that. And that's actually what I have more contestation with, which is those are the things that actually like drive me crazy more.
Yeah, well, I suppose on some level, science, like religion, is a form of prophecy.
Mm-hmm.
You know, and maybe a more reliable form.
Yeah, well, except that I would say that it, you know, religion has texts that I think are a bit more deified and less malleable and agile,
Absolutely.
and weren't written to have iOS updates like every day.
Absolutely.
Whereas I think religion has a bit more of like a fixed sense of the world that you either adhere to
There's
or
no
you.
final word of science,
Yes.
the way that there is a final word of God, and then
Exactly.
another one.
Yes,
You know,
exactly. Or or your interpretations of it. Yeah. Exactly.
And many of the mysteries of the metaphysical are actually patterned in the physical. So if you're really curious about trying to excavate some foundational cosmic intelligence, then look into the physical systems, because that's where that intelligence is patterned.
Yes.
And the more that you begin to understand about things like mechanism, the more consilience that you find across soil and gut. And yeah, you can overuse that poetically from time to time.
Yep.
But it is constantly fascinating to me when I, discover, a hormone in the body that has an exact counterpart, or this consistent sort of yin-yang that continues to arise, this sort of coincidentia positorum or whatever, that we can learn a lot about, maybe not about why we're here, but about how we're here.
Yeah, absolutely.
And that's the fascinating piece of science.
My question about it, I'm not a synastric person, but I also recognize how important pattern recognition is to people making meaning for themselves. It's not how I necessarily do it, meaning it's not the how that I do it, but I think where my skepticism creeps in sometimes. And look, I think we make meaning in all different kinds of ways. And by the way, there's scientists that are deeply religious, and we're all full of our own conflict and conflicting ideas. I think what I have had challenges with is more the lack of curiosity and the openness and the ability to recognize that by, like, for example, if you say to an astrologer, you're a pattern recognizer, right? There's a lot of it's like an insult, right?
Right, yeah.
And I think that's where I get hung up, which is that there's this inability to kind of zoom out on and look down and say, why am I making meaning this way? Why did I choose this way of making meaning? What about this methodology? Also, this methodology is based on another methodology called astronomy. Do I know anything about astronomy? Do I know what a moon is? Can a moon actually rise? Can the things I'm being told even happen astronomically? And a lot of times the answer is no. And so where I get caught up is this kind of wall that I've decided that astrology is this. I've decided that a Pisces is this. But with no further inquiry,
right?
And both self-inquiry as well as what is this based on? What am I'm making big life. I'm just using astrology as an example. Hope I didn't just create 25,000 enemies. I just mean it. It's with anything, right? Yeah. And I think even with science, you see this in a lot of ways where science, just going back to the beginning of our conversation, where bacteria has been otherized. It's And you see this from the last few years where all of a sudden science is big pharma and science is biotech and science is this and therefore it's bad. And therefore the people who are working on these things are bad and the people who do this are bad because I know, and this is how pseudoscience works and how Trump did this incredibly effectively, I know a secret that nobody else knows and I am the underdog for knowing it, and my value and my framework and my affirmation of the way I'm going to make choices can be reinforced by the fact that I so deeply know that I'm right, and the way, and that this thing that's being told to all of these people is wrong. Yeah. And I think, which may be true in some cases, like in some cases might be true. It might also be the symptom of really poor communication styles and skills. It might be a million things, but what science is, as I said, is a methodology for inquiry. Yeah. And what I really have problems with is that a lot of the ways a lot of spiritual things manifest is that it manifests with a level 1 question, maybe level 2, and then once you've like found your person, guru, answer, framework, it's like now you know the answers. And there's like I find that and even from people saying I'm a seeker, I'm this, and so I'm not saying everybody, but it is something that I have found and then have found challenging in speaking to people, then trying to bring up a conversation where they're just so sure when I'm not, like, I'm like, how, nobody's sure. Yeah. And I think that I find that to be the same otherization that we've been victim to forever.
Yeah. I think, where I draw a distinction is Taoism, that was very concerned with the study of nature.
Yes.
Now, not nature with microscopes the way we have it, sure, but certainly the course of nature, mm-hmm, and attributing much of the fundamental intelligence of the world to the way nature works. Yeah. And, or Buddhism, which really saw the world as mutually interdependent, mm-hmm, or have these concepts of dependent origination, mm-hmm, for example, yeah, where everything is dependent on everything else, and there's this beautiful image of Indra's
without
net.
attachment.
Without attachment, yeah. And I can't help but build that bridge between, that image of Indra's net, a mm-hmm, Buddhist image of this, yep, web that goes on forever with a diamond in each juncture, mm-hmm, that reflects every other diamond. I can't help but to build that bridge between those ideas. And then when I discover, like, oh my God, I've co-evolved, yes, over billions of years, absolutely, with these bacteria, mm-hmm, in my gut, and that there's this kind of co-tenancy situation, mm-hmm, and I am, there is a mutual interdependence, mm-hmm, and it perhaps that Gautama was more of a psychologist, mm-hmm, than he was a theologist, but I know that that's dangerous to make those conflations, mm-hmm, but
as I said, I think it's when there's a finality to it, and then a belief that that's how it is, and that you just know that's how it is, because yeah, I think what these things show you is like the diamonds reflecting each other. Like, we're going to continue to know more and, yeah, more and more. I think that whatever ways you can, actually think the connectivity and conflation is really much needed in the world, actually, because I think in some ways we've really siloed all of these areas in ways that really don't serve us. And I think that siloing and the lack of ability to make connection amongst these ideas is actually what has reinforced the division, and therefore really reinforced that, like, staunch belief that it's like this way or this way, even from the disciplines that say it shouldn't be that way. And so I think in a lot of ways, I think the more you can, I don't think what you're doing is making astrology out of microbiome. Yeah. I think in a lot of ways there's a beautiful practice of, metaphors are like some of the most powerful devices for change, right? Like, and I think where people can start to understand just the actual connectivity between all these things, the idea that 10 years ago we couldn't have even had this podcast. So, that and 50 years before that you couldn't have talked about certain other things, and so I think the embracing of like ideas that evolve and change and that deepen our sense of like knowing ourselves and knowing the world and actually finding ways to like connect these different ideas more. It's why we at Seed, like it's why we work with so many artists and poets all the time, is because you need to shift perspective. You know, that's where change is going to come from. I mean, I always wonder, like, you know, what would have happened in the last couple of years if, like, we just, I mean, it was like COVID the musical. Like, I just wonder what are, you know, you have to think about the mediums, and you know, Marshall McLuhan always talks about the medium is the message, and so I think the really big problem is just the fact that we have really ingrained ideas that whoever's delivering the message, we've conflated the who's delivering the message with the validity of the message.
Right. There are a lot of people that are holding on to faith because they either don't understand the substrate of the situation, or or there's someone that claims that they do, and it's easier just to put their faith into that demagogue, autocrat, etc. Sure. But then you have, you know, the other side, which is, okay, well, we've now invested all of our belief in science or in humanism, or, you know, we're going to instill the world with reason. And of course, that's where I generally lean a little bit more. But as a spiritual seeker, you know, I'm not sure that life is completely empirical, or could be understood completely empirically. Yep. So, you know, and this is why metaphor as heuristics can be really, really helpful.
Absolutely.
And I think Seed, for example, does such an incredible job with that, because, as I think I've told you before, when I open your website, I basically think I'm on the MoMA website or something. Thank you. I don't associate it, you know, with a microbiome company, but I know you guys are way more than that. Your vision is so expansive. But I think that the more that we can have some of these kind of conversations about science in a way that feels very poetic and has metaphor,
Absolutely.
I think the more that we'll be able to communicate it to a wider audience, and the more that we'll be able to understand each other.
Scientists want that too. I mean, we often do things with their science, and then they'll say, "Oh my God, like I never knew it could look like that," or be, you know, there's look, it's kind of like nutrition's not taught in medical school, right? It's you know, it's only now, actually Alan Alda created a beautiful program at Stony Brook about the community. He's on that 70s show that, I forget the name of it, that was
MASH.
No, for PBS.
For PBS.
He would like communicate really hard, like complex scientific ideas, and he loves science communication. I think actually his book on this opens with the George Bernard Shaw quote that says, you know, the greatest illusion of communication is the, you know, perception that it's like occurred. I think I made that, paraphrased it a little, but it's just this notion that, like, you know, Alan Alda has thought a lot about science communication. He established this beautiful program for science communication at Stony Brook. And now what you're starting to see in PhD programs, and even in other graduate programs and even undergrad, is starting to teach about science communication, because there's another beautiful quote that we love at Seed that we think about all the time, which is this notion that science isn't finished until it's communicated. And I think I really deeply believe that. And I think that, you know, communication, and I think what you see in some of these other disciplines and areas, particularly more spiritual and religious realms, is that the language and the medium and the aesthetic is beautiful. It's appealing to the human senses. It's delivered in ways that resonate deeply, spiritually, cognitively, intellectually, emotionally. But of course, the way science is delivered is, sixth grade bio was so boring, and I've never learned anything in science ever since then, because my teacher was boring and the textbooks were awful. but you think about like the way where you get to, I mean, look look where I could do what you created here with, like, I could do yoga, I'm sitting in nature. Like, there's really no reason why isn't this a PhD program? Why isn't, we don't have, there's not beautiful architecture to communicate. You go to a church, it's like you experience the aesthetic of some of these things, and it's like transportive. And I'm a New York Jew, and you walk into like a beautiful church in like Rome, and you're just like, how could I not be transported here? I mean, these are beautiful and aesthetic. So I think aesthetic, language, the lexicon, and the messenger are all the things that so many disciplines got right, and understood how to touch people in ways that, science just ended up becoming very medical, very clinical, very complex, very industrialized.
Yeah.
And I think as a result of that, we are where we are.
Yeah. I don't know if you've been going to some of the immersive art exhibits, like
Yes.
the Van Gogh exhibit or the Klimt exhibit, or there's a
Yes.
Frida Kahlo exhibit, and then there's some spin-offs, some of which are better executed than others.
Yes.
But oh my God, the seed exhibit,
Yeah.
the immersive microbiome
We're on it.
geodome experience.
Yes.
You're all
We're
over
on it. COVID was an interesting, obviously we had to really think about ourselves in pixels, which was hard, but obviously led to a lot of creativity. But now that there's some IRL happening, we've been thinking a lot about how this could show up in the world. So
Cool.
yeah, I agree with you. And I hope, and we think that it would feel quite spiritual as an experience. I mean, the Rain Room is a beautiful example of like collective experience of something that, is, and there's so many examples of them now. But yes, those things they're transportive, and they give you a new medium to talk about things that.
I could see it, the seed microbiome geodome
Yes.
at Lollapalooza.
100%, yes. We are very much headed there.
Yeah.
Yes.
All right.
Just give us some time.
Ara Katz. Hey, I'm glad you enjoyed this video. We have conversations with today's top thought leaders every single week, so make sure you click the notification bell below so you don't miss our weekly videos. I'll see you soon.